questions here. I'm all ready to go through our last unit that you'll need to know for your Biology Paper 1. So this is about B4 Bioenergetics, and hopefully it's quite a quick unit and it really is underpinned by just some key equations that you need to know and be able to apply in different scenarios. So let's have a look at those key equations to start off with. So here they are. These are the equations that you'll need to focus on and make sure that you learn. There is the photosynthesis equation, which is carbon dioxide plus water reacting to make glucose and oxygen. You'll need to know the word equation, that you need light energy in order for that reaction to happen. And you also need to know the balanced symbol equation. I always say it's a bit of a devil to balance this equation because you need a 6, a 6, and a 6 in front of all of those small molecules. Note the formula for glucose as well involves 6s and 12. So it's actually, if you think about 2, 6 and 12, all of those numbers are very important for these molecules. Aerobic respiration is photosynthesis backwards. So you actually need glucose and oxygen to produce carbon dioxide and water. And you can see that those symbols are exactly the same. And the way to balance the equation is again with those 6s in front of the small molecules and the glucose formula being C6H12O6. You also need to know about anaerobic respiration in both animals, bacteria, plants, and yeast. In an exam question, it will tell you if it's a plant or an animal, so you need to be able to work out which of these you will need to use. You don't need any formula equations for these, you just need to know the words. So anaerobic respiration is when you take glucose and turn it to lactic acid, and that's without oxygen, that's why it's anaerobic. And anaerobic respiration in plants and yeast is when you take glucose and it reacts to produce ethanol and carbon dioxide. So the point of respiration is really to release energy, and the point of photosynthesis is to basically produce the glucose molecule that we can then use in respiration to release energy. If plants didn't carry out photosynthesis, respiration would be not possible because all living things require the glucose made by plants in order to carry out respiration. So now that you know the basics of this unit, we're going to look at each equation in turn and see how that can apply to different scenarios. So let's look at photosynthesis first and have another look at that equation. It's really important that you do know that those products, glucose and oxygen, are made from the reactants, carbon dioxide and water. So just make sure that that is embedded in your learning. And photosynthesis, that reaction there, I'm going to put it in the top right-hand corner all the time of those slides so you can really focus on it. Because if you get an exam question on photosynthesis, the very first thing you should do is write out that equation. So you're really focused on exactly which equation that you're going to need to use to answer the question. And that equation shows you that photosynthesis is an endothermic reaction. The light energy going into the reaction means it's endothermic. So energy enters. And it takes place in the chloroplasts of plants, which is why only plants can carry out photosynthesis, because it's only them that have those chloroplasts. And inside the chloroplast is a chemical called chlorophyll, which is a green pigment or a green colour, and that captures the light energy in order to turn the carbon dioxide and water into the glucose and oxygen. So without chlorophyll present, you couldn't capture the light energy. So a lot of questions are dependent on that knowledge of chlorophyll being present, therefore photosynthesis can take place. Oxygen produced in photosynthesis is actually a byproduct. That means not all of the oxygen is going to be used by the plant, and most of it leaves through the stomata. And then it enters the atmosphere, and animals can then breathe in that oxygen. They don't do it to help animals. They are doing it because they need the glucose for the plant themselves to grow, and the oxygen is a byproduct that we can use. There is a required practical to do with photosynthesis. Again, really write out that photosynthesis equation so you're really focused on exactly what is happening in anything to do with plants and photosynthesis. So the required practical that we do uses pondweed, and pondweed is an underwater plant, and actually it's a really useful way of measuring the rate of photosynthesis because you can observe the bubbles it produces when it photosynthesizes under the water. You can change the effect of light intensity and measure the number of bubbles of oxygen per minute. The light intensity in that situation is the independent variable. That's what you're going to change. The number of bubbles of oxygen per minute is the dependent variable, and that's what you'll measure. So this is how you carry out that required practical if you were going to investigate light intensity and how that affects the rate of photosynthesis. So first of all, you cut the piece of pondweed at a required length. So you could say 10 centimetres, that would be a control variable. You would then place the pondweed in a tube with a specific volume of water, making sure the pondweed is completely covered. Again, that is a control variable. And you'd put the pondweed in the tube inside a beaker of water. Now, the beaker of water is there because you might get some heat released from the lamp, and that could affect the rate of photosynthesis. So again, using that water bath with that clear beaker allows the light through, but helps you to control the temperature of the water. So as a control variable. Then first of all, you'd put your lamp a set distance away. So you'd set up a meter rule next to your beaker and place the pondweed around 10 centimetres away from the lamp just to start with. Leave it for 5 minutes to acclimatise and get used to the light intensity, and then start recording your data. So you record the number of bubbles of oxygen, for example, in 1 minute. That would be a dependent variable. And then you'd start to change the independent variable by moving the lamp further away, say 10 centimetres away, and then recount the number of bubbles per minute and repeat that up to 1 metre. You'd then have to repeat the whole investigation again a further 3 times in order to collect some data which is valid, which you then use to calculate a mean. Once you've got that data for that investigation, you can use that to compare the effect of light intensity on the rate of photosynthesis. So you've moved the distance of the lamp and seen how that affects the rate of photosynthesis, because the further the lamp is away, the less the light intensity is. But you can also use that equipment and that method to change the temperature. So, for example, you could change the water in the beaker, keep the distance of the lamp the same, that becomes your control variable, but just alter the temperature of the water in the beaker each time and see the effect of that on the rate of photosynthesis. You can also change the carbon dioxide by using dissolved carbon dioxide, so usually a carbonate solution, inside the test tube where your pondweed is. Or you could change the colour of light and see how that affects the rate of photosynthesis. So obviously, does that affect the absorption by chlorophyll and therefore the rate of photosynthesis? So all of that simple equipment and that experiment can be used to measure any of those independent variables. Whatever you change, everything else needs to stay the same and becomes the control variable. So, for example, if you change the light intensity, what you'd find is as light intensity increases, so that would be getting closer to the pondweed, the rate of photosynthesis increases. If you change carbon dioxide concentration, and that increased, the rate of photosynthesis would increase. And as temperature increased, the rate of photosynthesis increases. That's up to a point. So whenever we have a chemical reaction with temperature, the molecules collide more, and that's included in the photosynthesis reaction. So you've got more chance of carbon dioxide and water having a successful collision and reacting together inside those chloroplasts. However, photosynthesis is an enzyme-controlled reaction. So with temperature, it's always to be aware of, as the temperature increases, it will get to a point where the enzymes will start to denature. And therefore, the rate of the reaction actually drops away because it is an enzyme-controlled reaction. But if you look at that equation and you use that equation, it can help you to work out what you might predict from changing things like carbon dioxide. Because if you put more carbon dioxide, which is a reactant, into the reaction, you'll get a higher rate of photosynthesis. Now, if you're doing the higher paper, you have to be able to refer to those ideas and talk about limiting factors. If you notice this graph, you can see as the rate of photosynthesis is up in the vertical column, and then horizontally you've got the light intensity. Lumens is often the unit given or used to measure light intensity. And you can see as you increase the light intensity, the rate of photosynthesis increases. So at that point in that graph, the light intensity is actually limiting the rate of photosynthesis. When you get to the maximum rate of photosynthesis, it levels off. This means that it doesn't matter how much more light intensity you put in, there is another factor limiting the rate of photosynthesis, and that could be carbon dioxide or temperature. Carbon dioxide, temperature, and light intensity can all limit the rate of photosynthesis. Water isn't on there, and you might expect water to be on there because water is often in excess in a plant. There's other things that will go wrong before photosynthesis is limited with the amount of water plants need. So that's why carbon dioxide temperature and light intensity are all really important factors in limiting the rate of photosynthesis. If you increase one factor, the others can become the limiting factor when you reach that maximum level. Another way of looking at this is to think about the economics of enhancing conditions inside things like greenhouses. So if you can understand limiting factors, you can maximise the profits for any farmers and maximise the rate of photosynthesis. So for example, in that greenhouse on a hot summer's day, if the temperature is warm, that won't limit the rate. If it's summer, there'll be lots of light, that won't limit the rate. So the rate of photosynthesis might be limited by the carbon dioxide concentration inside the greenhouse. So you might need to open windows and try and get more ventilation. On a cold winter's day, the rate of photosynthesis is most likely to be limited by the temperature because it's cold, and in winter there's less light, so temperature and light would limit the rate. In that case, to maximise the rate of photosynthesis there, you'd need artificial lights and something like a propane heater that will release heat and also releases carbon dioxide because of the way the combustion reaction works. This carbon dioxide can then be used by the plant for growth. If you're doing the higher paper, you also need to know about the inverse square law. And this is a way of interpreting the distance of the light source to light intensity. So the inverse square law basically says that light intensity is inversely proportional to the square of the distance from the light source. And that can be summarized in this equation here, where it says light intensity proportional to 1 over the distance squared. That basically means if you've got those two bits of information, the light intensity and the distance, then your graph would actually start to look like this. If you looked at the rate of photosynthesis, it would slope down. Okay, so it basically means that if you're doubling the distance, the light intensity will decrease by a quarter. And there's this quite nice diagram here where you've got the light source and you've got a given area. And as you double the distance, you can see the light intensity will decrease by a quarter because it's spread over much more area. You can see that as a value as well. If you're calculating this, you would have to work out the distance and square it, and then you do 1 over distance, and you can see with the values that you get as you double the distance, and that's doubling, remember, it's not going up by 5s, it's going from 5 to 10 to 20 to 40. So as you double the distance, then this quarters each time. One of the last bits about photosynthesis is to realise what the plant actually needs the glucose for. So the plant needs the glucose for its own growth. And part of that is to make different molecules that the plant will need in order to function properly. So a useful way of remembering this is the acronym CROPS. So CROPS, obviously you grow crops, they are plants, and it also stands for these common things that plants need to make. C stands for cellulose, which is used in cell walls, and that's used to strengthen the cell wall. R for respiration, that is aerobic respiration as well as anaerobic respiration. So you need glucose as your raw material for that. O for oils and fats, in other words, lipids. So fats and oils needed for storage in the plants to make their seeds. P for proteins. You also need nitrates, remember, from the soil to be able to produce those amino acids that make proteins. And proteins are essential for growth. And S for starch. Starch is a large storage molecule which is insoluble. So it's always a source of glucose for the plant should it not be able to make glucose by photosynthesis. And all plants carry out photosynthesis and respiration. All living things need to respire. But photosynthesis only happens in plants or algae when there is enough light. So at night time, plants tend not to carry out photosynthesis. Their stomata close, remember, so they can't get the carbon dioxide and water in that they need to photosynthesize because there's no point in having them open if there is no light energy for that to happen. But the plants need to still respire on a night. So actually the amount of carbon dioxide produced increases. Whereas normally in the light, then when they're carrying out photosynthesis, the amount of oxygen is much higher than the amount of carbon dioxide that they produce by respiration. This brings us on to aerobic respiration, and this is the equation for aerobic respiration that we looked at the beginning. You can see it's glucose plus oxygen react to make carbon dioxide and water. So now this is the opposite to photosynthesis. So you can see it's exactly the same molecules, but the reactants have now become the products. It's not a reversible reaction because this happens in plants and this happens in animals, but animals cannot photosynthesize. It is two separate reactions, but they actually use the products of one are used as the reactants of another. And if we have a question on aerobic respiration, you need to have that equation of glucose and oxygen making carbon dioxide and water in your mind, and you need to write it down for the question before you start to answer it, just so you're really focused on, am I using the right equation? Is this the right way around to say things? Am I talking about the right things? And really focus on what that means. So aerobic respiration happens in the mitochondria of eukaryotic cells, and its purpose is to release energy needed for living processes. In doing so, it's an exothermic reaction. So you also get heat energy released. It is also controlled by enzymes, just like photosynthesis. So you've got the same requirements for temperature being pretty stable. So even though this reaction produces heat, that's got to be managed by the animal or plant that it's doing respiration in, because if the temperature gets too high, the enzymes will start to denature. And the cells need that energy for things like cell division, movement, for keeping organisms warm, processes like active transport, and for chemical reactions to build larger molecules such as proteins as well. So you can see you'll need respiration to happen all the time to make all those specific things happen, as well as many others. Anaerobic respiration happens when oxygen is not present. And this doesn't take place in mitochondria, it takes place in the cytoplasm of cells. In animal muscles, in particular, you'll need anaerobic respiration to keep those energy requirements going when you exercise and things like that. So the reaction, which is anaerobic respiration, taking place in cytoplasm of muscle cells, would be glucose is converted to lactic acid. You don't need to know the formula for the lactic acid molecule. If you're exercising, then anaerobic respiration will start to take place in your muscles. Now, this is a backup, if you like, from aerobic, because much less energy is released than aerobic respiration, because what we say is that happening is the oxidation of glucose. And in this reaction, it's incomplete, but in aerobic respiration, it would be complete. This incomplete oxidation of glucose causes the lactic acid to build up and creates something called an oxygen debt, and that's the need for extra oxygen. During long periods of vigorous activity, your muscles will then become fatigued and stop contracting efficiently. So it's the oxygen debt coupled with the lactic acid that you're producing in your muscles that will stop them contracting efficiently and becoming fatigued. When you do exercise, though, the body usually responds in a way to increase the amount of oxygen and glucose being transported to cells. So you try to avoid anaerobic respiration if possible. So during your exercise, the human body increases the heart rate, the breathing rate, and the breath volume in order to get more oxygenated blood. This allows more aerobic respiration to happen because this blood is transported faster around the body, carrying more oxygen for aerobic respiration. This allows more energy to be released and it allows the contracting muscle cells to continue to contract. If more aerobic respiration is happening, more carbon dioxide is also produced. That can be transported through the blood plasma to the lungs where you breathe it out. So in the response to exercise, you really need to remember that aerobic respiration reaction, because you can see that the glucose and oxygen need to be increased. The carbon dioxide also increases in order to release more energy And that's why we need all these transport mechanisms happening faster and more oxygen in your blood. If you're thinking about the glucose not just being transported but running out of glucose, you don't actually run out of glucose because you have glycogen, which is a glucose storage molecule, in muscles and liver cells, which can be converted to glucose rapidly. So you don't actually need to take in food while you are exercising because you have a store of glucose in the form of glycogen. If we just think about exercise then generally, if you're working hard and exercising your muscles, we talked about aerobic respiration being our first primary way of releasing energy and anaerobic respiration as a backup. If you're doing the higher tier paper, you need to focus a bit more on the oxygen debt side of things, the things that can happen when you're exercising to make you have an oxygen debt. So if you think when you first start exercising, your oxygen requirement rapidly increases. You temporarily are in oxygen deficit until you start to have an equilibrium where your oxygen intake matches the aerobic respiration. When you end your exercise, though, because of that initial oxygen deficit, you've actually got oxygen debt. And you also have oxygen debt from when your lactic acid has been produced. To convert the lactic acid back into glucose, you need more oxygen. So as you can see, the blood flowing through the muscles transports your lactic acid to the liver and is converted back to glucose there. And that requires oxygen, and that is really why you've got oxygen debt. So oxygen debt is really the amount of extra oxygen the body needs after exercise, and it needs it to react with the lactic acid and remove it from the cells. So that's why we call it oxygen debt. And the last little bit starts to look at anaerobic respiration, but not in animals, in plants and yeast. When anaerobic respiration, remember, happens when oxygen is not present and takes place in the cytoplasm of cells. That's true for plants and yeast, as well as animals and some bacteria. So the reaction can be represented as the following. In plants and yeast, they take the glucose, they react it to make ethanol and carbon dioxide. And this simple bit of equipment here can show you how yeast cells in a sugar solution, if you put them a layer of paraffin at the top, it will stop oxygen getting into the yeast cells, so you're forcing them to anaerobically respire. So you can see that carbon dioxide bubbles are going to be produced, which turns lime water cloudy. Those yeast cells will also be making ethanol, but that will stay in the solution. Anaerobic respiration is actually a type of fermentation. So it's called fermentation as well as anaerobic respiration in plants and yeast, and it's got economic importance in the manufacture of bread because the carbon dioxide made helps the bread to rise. It's also important in the alcoholic drink industry because it produces ethanol. So you can see to make beer, the fizziness of the beer comes from the carbon dioxide and the alcohol comes from the ethanol produced by the yeast. The ethanol, the alcohol in the wine, comes from the yeast making the glucose and turning it into ethanol. The carbon dioxide tends to go into the atmosphere in that example. And in bread, to make it rise, the carbon dioxide produced helps to make air spaces in the bread. The ethanol actually evaporates when it's cooked. One last little bit looks at all of the reactions in our bodies. Now, this links in really to all the units we've covered so far in paper 1. Metabolism is a term that we use to talk about all of the reactions inside a cell or the body. And the energy transferred by respiration actually helps to make energy, release energy for the metabolic processes. And they're often enzyme controlled. And when we think of metabolism, we are thinking of things like the conversion of glucose into starch, which happens in plants, or conversion of glucose to glycogen in animals, or the conversion of glucose to make cellulose in plants. We're looking at the formation of lipid molecules, and you should know they're made from a molecule of glycerol and 3 molecules of fatty acids, because that in the digestive system is what lipid molecules are digested into. So that's what they are actually, we need to make them as well. Glucose and nitrate ions actually form amino acids, and they're used to synthesize proteins, and we've again looked at that before. Metabolism also includes the process of respiration itself, but it also includes the breakdown of excess proteins to form a chemical called urea, and urea can be quite actually toxic if it builds up, so we need to excrete it. So, as a little quick summary of this unit, if you really do focus on knowing these equations properly, you should be able to apply them to many different questions about this unit. So I would heartily recommend that when you are practicing exam questions in the build up to your exams, you really focus on, is this question about photosynthesis, aerobic respiration, anaerobic respiration in animals or some bacteria, as an example they've given, or anaerobic respiration in plants or yeast. And the first thing you do is write down the word equation. So you're really focused on using exactly the right words in your answers. Okay, that's it for now. So good luck. And I hope your exam goes well. I've got the 4 videos for you to go over and have a look at. So I suggest you quickly run through them all. If it's the night before your exam, then best of luck. But if it's earlier than that, then you've got plenty of time to go and look at this a bit further and do some more practice questions, or obviously the Quizlets. Okay, thank you. Take care. Bye.